Welcome to the BioRender 101 webinar. Um, my name is Shiz. I'm actually one of the co-founders, and in my previous life was a medical illustrator. So happy to answer any questions about medical illustration in general, figure making. I'm going to be going through some basic tips of how to use BioRender specifically, but along the way, naturally, we're going to cover some design tips. So hopefully you'll walk away from this webinar with some new tricks in your back pocket, even if you're a seasoned BioRender user. Actually, we'd love to see show of hands if you can use a little raise hand function if you are brand new to BioRender. We'd love to see. Got lots of hands raising here. I like the Zoom function. I don't have to look at Zoom to see how many hands are raised. So quite a few. That's great. Thank you. Thank you for participating. If you can see my screen, I am on a Chrome browser and I'm on the BioRender sort of login page. Now, this is a little bit different from the BioRender, website, I guess, homepage, but you can navigate to this sign-in page through the biorender.com website. So just to differentiate those two, sometimes people get confused. You can click here to sign in, and then it'll take you to this exact same sign-in page. So if you already have an account, go ahead and log in with your credentials, but I'm going to use my demo account. Right. Oops. And we recommend using Chrome just because it's the best browser we find for these sort of graphics heavy apps like BioRender. But we do support other browsers like Firefox, Safari, Edge, I believe, if you're on Edge. All right, so here's the BioRender interface. Some of the feedback we've gotten is it kind of reminds them of Google Docs, a visual version of Google Docs. And it'll obviously look different depending on everybody's accounts. Mine is a little messy right now because I've been really busy in this app, but I've got lots of different types of figures and different formats. So we've got, slide presentations I've made in BioRender. I've made posters, cell-to-cell interactions. I even have my own little activity sheets and design tips as I'm doing webinars myself. So just getting a little bit acquainted with the user interface here. These are all my figures in this section below. On the left-hand panel, we've got sort of a, I guess, folder system, you could call it. There are images and folders shared with me. For example, maybe a teammate has made a bunch of figures, they've thrown it into a folder and they've shared that with me. That's showing up here in my left-hand panel. This drop down are all the figures and folders created by me, and I haven't shared it yet. So it's not showing up in, a shared context at all. It's just me privately. If I navigate up here, we've got our big BioRender templates tab with all of our beautiful pre-made templates. A template is just a pre-made figure. Basically, it could be anything from a simple, 4-step sequence like this, that's blank, all the way to a nearly finished figure. So sometimes people like to start here if you want to type in something specific. Let's see, PCR. If you want to show sort of like a protocol, you can type in PCR, see if anything comes up here that's of interest to you. If you want to get more specific, maybe you want to type in, a machine that you're using in a PCR process. You can also sort by or search by category, but I would recommend using the search panel up here. If you're inclined, you can try out our new posters feature. I'm not going to go too much into detail of this, but just know that we do have a really snazzy poster maker in BioRender with, columns and rows, that sort of format when you stretch and skew them, which is really, really cool. Let's go back to the homepage here. Again, my figures below. Here's another sort of sampling of 1, 2, 3, 4, 5, 6, 7 BioRender templates. We're just sort of presenting to you as options. And again, if you click this button, you'll get sent to the whole library of templates. But I'm going to go ahead and start with a um, brand new figure. I'm going to go here to create new illustration. And I'm presented with this sort of blank canvas. And just to get a little bit acquainted with this interface, we've got a lot of options here on the left-hand panel for icons. We've got templates, brushes, favorites, uploads, all of that. What happens up here in the nav bar or navigation bar are more options that will pop up as you add icons. So for example, if I add in, say, a cancer cell to my canvas, I'm going to drag and drop. You see that when I've added that, I've got a whole bunch of new options that pop up here, as well as new options that pop up on the left. This is, I'm using a paid version of BioRender right now, but for the most part, a lot of the features are the same. I'd say a few key features you get access to when you unlock and use the upgraded version of BioRender. Lots of cool features like slides, for example. I can make an entire slide deck in BioRender so that my next presentation is very visual. And a couple of other things like layered icons, gradients, higher resolution export, things like that. So I'll call those out as I go along of whether it's available in the free version or not. I'll try my best to differentiate. So here's my cancer cell. Obviously, lots of options here to change the color of that icon. A lot of pre-colors here, preset colors. You can also change this cell to pretty much any color under the rainbow and any saturation or dullness, which is really cool. I'm going to apply that. And that's sort of like really getting started. A blank canvas, you can drag an icon, edit that icon, and then what you do is you just sort of build upon that, add more icons and more elements. I'm going to go back to my homepage though, because I've got a couple of style tips here that I'd love to share with you. I've got my header slide to kick off our demo. And again, this is using the upgraded version of BioRender. You can see that I can hide and show my slides here on the right-hand panel. And digging into BioRender, I know a lot of us are coming in with ideas of how you want to use BioRender, probably a certain use case in mind, like a presentation you have coming up. Maybe it's a lab meeting, or a big presentation you're giving, or just one that you're giving in a small meeting with a couple of colleagues. Always good to have some visuals to back up your stories. You might be preparing for a publication, in which case you might be making a graphical abstract or any of the figures within the body of the text. You might be making a poster. We're all going back to conferences now, so poster, again, you can make the entire poster in BioRender or just the figure. But I do want to highlight a couple of use cases that might not be immediately evident that you can use BioRender for, and that we're hearing more and more feedback that it's becoming really critical in scientists or teammates day to day. Whiteboarding, as I mentioned, lab meetings, grant proposals. You can use the free version for grant proposals. Obviously, I'd recommend upgrading for the high resolution export, so you can have a high res, crisp export to include into your grant. Process diagrams, experimental protocols and guides, even social media, onboarding materials, and many, many more. I'm sure many of you can come up with your own ways to use more visuals in your day-to-day, but these are the common ones that we hear feedback about. Okay, so just to get comfortable and acquainted with the different types of icons in BioRender, sometimes that's feedback we get as well, is that there's a lot of different things you could build in BioRender and many different ways to do it. So I thought I'd sort of show you the different types of icons. You don't have to memorize this, but just to get acquainted with, again, the types of icons we have. So if I type an antibody here into the search bar, you could type in anything if it's related to your research. Maybe it's a type of mouse strain, maybe it's a type of machine. I'm going to use antibody for now. And you can see all the different types of antibodies that show up in the search results. Some are a little more complex than others. I'm going to just drag out this simple antibody or representation of an antibody. It's blue. If I zoom in, you can see a slight outline around it, very clean representation. Awesome. So this was what I would call a regular icon. Regular as in it's sort of static. It's static, but you can, change the color of it, sort of like putting a color overlay. So the original was blue, but maybe I want a red one, or maybe a fluorescent glowing antibody. So there you go. You can make that regular icon glow. But most icons are quote unquote regular icons that behave like this. Now, a regular icon with layers, I would say maybe something like a cell is a good candidate to call a layered icon. And the reason being is we've actually extracted, I guess, the layers of this icon for you to be able to independently edit it. So say you want the nucleus to be lighter or darker, or maybe you want it to have a corrugated edge around the nucleus if it's sort of degrading during apoptosis. You can actually do that now in BioRender. So I've got my green cell, and I'm going to come up here to layers. Now, not every, regular icon has layers, but some do. That's why I want to really highlight the ones that do. And you'll know if it does or not just by clicking on it and expanding this layers panel. So here I'm on the cell layer, or I guess the membrane in this case, or the nucleus layer. Let's click on the nucleus layer and see what we can do with it. You can change the transparency, so make it disappear completely, or make it visible, or make it a little bit more transparent. This is actually really helpful if, say, you're adding a label like T cell, and the presence of the nucleus is a little bit distracting, but you don't want to remove the nucleus completely. Oops, where's the nucleus layer? There we go. You can actually make the nucleus a little bit more transparent so that, the T cell label shows up a little bit better. That would be one use case for making this more transparent. Saturation, if you want it to glow, or maybe you're staining the nucleus with a certain color. You can do that as well. You maybe want to then make that fill color a little more fluorescent. Oh, it's low transparency. That's why it's not looking very glowy. There we go. So you can see here, I've made the nucleus glow. I can also make the borders of that nucleus dashed. Now, again, this is a cell, but you can see how you can use this for, say, like, I don't know, a mouse tumor, or a protein. Manipulating the edges of the icon might be something you'd want to do. And this is how you would do it. Cool. Okay, so now this looks like a T cell with a nucleus that's glowing, where the nuclear membrane is sort of degrading, or maybe porous. But that's important for you to show things entering and leaving the cell. Okay, so that's a regular icon with layers. Grouped icon, probably self-explanatory, but just in case we're not on the same page, I'm going to type in antibody again. And then you can see that some of these icons that show up, there's a little purple bounding box that shows up if I scroll over some of them. Those mean that it is a grouped icon. Here, let me zoom in a little bit. If you can see here, this purple box that shows up as I scroll, these are grouped icons. And basically what that means is our design team has gone through and sort of layered and grouped regular icons into a grouped icon. So it's easier for all of you to have another starting point. Maybe it's a cluster of icons like this. So you can imagine if I double click, I'll probably be able to, independently manipulate some of these icons within the group, and then go ahead and edit that single icon as if it was a regular icon. But if I double click out of that, I'll have that group to manipulate again. If you use Illustrator or Photoshop, it sort of works the same way. But that's an example of a grouped icon. Another reason why you'd want a grouped icon is maybe our artists have put together an icon that is even more manipulatable. For example, this antibody with these sort of markers on them on specific domains here on this region of the antibody. Those can be removed. I think these are even shapes So if I click in and click in, these could be like edited or manipulated even further. And these aren't actually made of regular icons. These are made up of shapes. See, my options have changed here. It looks a little bit different. It's because these are BioRender shapes. Sort of in PowerPoint or Illustrator, when you make objects with shapes, it's not a flattened icon. It's more of like a, circle, square, star sort of a shape. And then obviously you can like edit those individual parts as you see fit. Maybe you want to highlight the farmost domains like this. Anyway, that is an example of a grouped icon. Everything looks a little bit hazy here and light gray because I'm still in sort of what's called an editing mode of this grouped icon. So if I click out, you'll see everything kind of comes back to full opacity. And then you can have groups within groups within groups. So just be careful if you end up in an infinite spiral of grouped icons. You can usually avoid that by going into the, I guess, layers of groups and ungrouping them. But that might be too much information for now. Let's just keep it as a grouped icon is a set or a grouping of regular icons. Now, brush icon, also pretty straightforward. I don't think we have any antibody brush icons, but likely you might need a brush icon when you're dealing with DNA. So I just typed in DNA into the search bar, and then I had some icons show up that were regular icons. So all these appear. Some that are grouped icons, like this one here, and some that have this little brush icon, and that's telling me that this is manipulatable. I need a better word for that. Editable like a brush. So basically, sort of a flexible, bendy pattern. And this has been important for a lot of scientific concepts that, again, have long strings of patterns, like maybe a membrane. If you're talking about the surface of a cell, you might need to show, waves or undulations in the phospholipid bilayer. So this is when a brush might be helpful. Now, if you're interested, I'm going to get a little bit technical here, but if you want, you can even expand this brush so that each little piece of this membrane is editable in and of itself. And that option is somewhere here. Separate brush into editable icons. Okay, so it's on the left-hand panel. I'm going to click that button and it's going to say, are you sure? Because doing this is not reversible and you're going to be able to edit, each little piece. Confirm. BioRender is very nice in that it has made it into a grouped icon so that I don't have a million pieces to move around. So I'm going to double click into this newly made grouped icon. And you can see here that now the phospholipid bilayer is sort of manipulatable, editable independently. Again, this is not a reversible action. So now this is permanently going to be, piecemeal like this, but you can always redraw it and very quickly edit those yourself. Okay, so that is the brush icon. Last but not least here is a PDB icon. Now, if you need a little bit more detail in this antibody structure, maybe you are a structural biologist or you have a favorite protein with the exact PDB ID, you can actually upload your PDB file or retrieve it from the PDB library. So I'm going to click this PDB retrieve. I think 1HZH is the one for IgG. There we go. So this just pulled right in from the protein data bank. And I can manipulate this to whatever view or vantage point I like. And why I would use this, sometimes you just want a bit of a pop of 3D or some ribbon or, quick surface view of the protein just to bring focus to your graphic. Shading style. I know this cartoon style is very popular. There's toon, flat. Anyway, you can play around with it and color by chain and whatnot. There we go. And I'm going to save as a new icon. So even though this is also a antibody, completely different shape, of course, and different purpose. Maybe you need a little bit more 3D structure to your diagram. So just wanted to highlight a few different types of icons in BioRender. And there are a few more, but they're not really icons. I'd say they're, picture types. And that is, uploads. You can upload anything into BioRender as long as it's sort of a JPEG or a PNG or an SVG file. Maybe you made your own icon in Adobe Illustrator and you want to bring that into BioRender to use. Some of you are very crafty. So you have your own icon set already. You can actually upload those into BioRender and then add it to your icon set as you're making a figure. Okay, let's move on to one tip about aligning objects in BioRender. We get this a lot. I don't know if this just is something in the science world where everyone loves to make sure everything's really aligned and straight. I'm definitely in that camp as a medical illustrator. I like things to be nice and neat. But I guess particularly helpful when you're trying to align panels and such for publication diagrams, or if you have objects like this that you sort of want to have in a straight line, and it's a little bit tricky unless you have the right tools. So say you have a bunch of stuff like this, maybe 4 antibodies in a row. There's different ways you can align in BioRender. One is grid lines. Grid lines is sort of like using graph paper, so I can turn it on and off here. If you can see, I just clicked this hide grid, show grid button. So you can use that just to kind of eyeball it. If you don't like things that snap in place, when you're using an app and then it sort of forces you to snap it into a certain place. Some people don't like that. So if you want to really freehand it, you can use the grid paper. You can also put down your own lines by coming up to the ruler. You can see here, I'm in the ruler area of BioRender. If you don't see this, it probably means your ruler is turned off. You can turn it on by clicking there. I'm just going to lay down a couple of guidelines. There you go. There's one. There's another. These are temporary little lines that you can put down yourself. I like to use guidelines to create a border around my entire figure like this. So it's sort of a visual cue to avoid putting anything in that area of the figure because many reasons, really, you just don't want things to kind of butt up against the edge. And it really helps to kind of create a visual border or breathing room around figures when you have a really nice buffer. And of course, you can hide these by right-clicking and hiding guidelines. Another way to align objects is to enable alignment. So again, if you're the kind of person that likes when things snap into place, you can go to View and Enable Alignment. So you'll see here then these objects will start to sort of snap into place. And my mouse is sort of forcing me to find these sort of places where it'll lock into place like that. I think PowerPoint has this, a lot of other features or products have this, but it's telling me these purple little lines tell me the distance between them. Anyway, I think you get the idea. And then finally, you can actually just use the align feature, which a lot of people don't know we have. Align, you can distribute evenly here. So I'm going to align everything with one button. There we go. And then I'm going to distribute them evenly. There we go. So that's probably the easiest one to use if you really just want something to be in a row. Numbering protocols, that's a good way to use this tool as well. Okay, so that's alignment and BioRender. That's a lot. So I'm going to move on to one other use case, which is very common, and that is protocols or timelines. If you're running an experiment, you want to show what you've done visually instead of just, a bunch of text on your presentation slide or your protocol section or method section of your manuscript. I'm going to type in protocol. And then you'll notice here that there's 2 little tabs when I use a search bar in BioRender. Icons show up related to protocols. So, machines, some vials, not really sorted by relevance though. just sort of a dump of icons. In this case, because protocol is more of a concept than a specific icon, I'm going to navigate here to templates. And now this templates tab is actually the exact same content you'll find if I go back to, say, my gallery and then BioRender templates. If I type in protocols here or protocol, the exact same search results will show up as if I was within that other view, this view. The only difference is it's just really teeny tiny when I'm working here in the search bar. So if I type in protocol, hit enter, and I go to templates, see, it's really small. Actually, if you roll over it like this, an expanded view will show up. So that's really nice. I'm going to go ahead and select this template. I just clicked it. And then I can either replace my current canvas or add to it. I'm going to go ahead and just add to my illustration. Okay, so you see here, basically added as if I've done the figure, which is great. Hopefully we have something close enough to what you need that you actually almost won't need to, make a figure in BioRender if we've got exactly what you need. Hopefully, in the best case scenario, we have what you need and you just kind of tweak it a little bit to your own liking. And you can, export, throw it into your slide deck or use BioRender directly to present, whatever you need to do. In this case, it added to my canvas, and you'll notice again that purple box showed up. So it actually imported as a grouped icon. We learned about grouped icons a minute ago. It's basically a glorified grouped icon. So what I can do, if I don't like that it's moving all in one chunk, I can come down here and edit the group. I'm actually going to click ungroup up here, ungroup, and this is going to expand this whole giant grouped icon into little pieces. So you see here, it's now no longer a grouped icon. And that's just easier for me to edit now. This looks great. I think maybe I don't like this machine. So I'm going to look for a different MS machine, mass spec. I'm still in my templates tab. I'm going to go over to icons, lots of different mass spec machines. Maybe it's this one that I actually use for my experiment. So I'm going to replace that icon. There we go. And then, yes, I'm going to run an analysis. And then maybe this is not the graph I want to use. Sometimes you want to actually add real data or findings to your diagram. You can actually upload an exported diagram from GraphPad Prism. So this is an example of that. I clicked upload here, and then upload again. And it's going to prompt you to grab a file from your desktop. And as long as it's, as I said, a PNG or a JPEG file or an SVG, we are, and this is a bit of a hint, we are working on improving the graphs feature. So there might be a world where you might be able to actually create a graph in BioRender soon, but not yet. Currently, you'll have to just import a PNG file that you've exported from GraphPad, which I've done here. So I'm going to go ahead and use that as my last step. There you go. Okay, so that's how I would, manipulate a template that is pre-existing to my own unique use case. And again, protocols or timelines are a very common way to do that. I also wanted to show, something like a timeline. I know a lot of common use cases are, say, using animal models. Maybe I'll use timeline mouse as a search term. And that looks pretty good. I'm going to go ahead and add to my illustration. Again, it's a sort of a grouped icon. I'm going to go ahead, I'm going to go ahead and ungroup it. And what I like about BioRender is how all of the icons sort of piece together really nicely. So if something's unique about this mouse, by the way, I just option click dragged to make copies of an icon. So you can do that if you're trying to make, a cytokine spray or something, or what's the word? Duplicate an icon really quickly. You can do that. But say I want to inject what I've extracted from one mouse to another, I can always change, the color of that mouse, like so. And speaking of layers, again, I'm using a mouse as an example, so apologies if you don't use mice, but a lot of our mice and special icons actually have the ability to be edited further by, by fur, by flesh, by eyes. If you want to change just the fur or maybe just the flesh, I guess, to be highlighted, you can do that. And looks like I can't drill down any further, but there's a way to do that as well if for some reason you need to color code parts of the mouse. You can also add things like, maybe a tumor. This poor little mouse might have a tumor. So if I type in tumor mouse, might get some options for a tumor. I'm gonna stick that on their back. And hopefully I have a color that sort of matches it. And then same thing goes for, I'm gonna option click, duplicate. Same thing goes for, any other colors we have, like so. All right, and that's really nice. Again, all the icons are meant to be worked together because you want it to look like one artist has created the entire figure instead of a mishmash. Sometimes what happens is we take graphics from Google search images and they all kind of come together looking a little bit like a Frankenstein figure. So to avoid that, we've made sure that all of our icons sort of play really nicely together. And this is an example of that. Some of our icons are actually even more editable than you think. So something like a syringe, if you need to show, parts of a syringe, you can actually, and again, you have to look for the sort of grouped icon label, but if you need to show something is, pulled back like this, in the case of a syringe, maybe you want to hide part of the needle if it's sort of being injected like that. Lots of ways to get creative with showing action in your protocols or experiments. All right. So that's protocols and experiments. I'm going to pause there for a second because I've spoken a lot. We're coming up on time shortly, and I'd love to see if there are any questions in the chat here. Lots of great questions. Thank you for them. Oh, okay. Yeah, adding elements like numbers to tubes and objects. Very good question. One thing I'd like to highlight is how quickly and easy it is to add numbers and sequences to steps. So, you have like steps of a procedure or steps of a figure or steps of a cellular process. I love using this insert shape and then these little pre-made circles with numbers inside it or letters. I'm going to select this and hit the 1. And then wherever I click down with my mouse, it literally just placed a perfect circle with a number inside. And that's very specific to these sort of, sequential type of diagrams. Because you notice that everyone was creating, a number and then putting a circle around it. And so this kind of makes it perfect that way. I'm going to option click and duplicate that. And then double click to make that a 2. Same thing, 3. If you're trying to label glassware, I think is what the question is. That's another really interesting use case. So let's see here if I can type in maybe a beaker or let's see, a test tube. And feel free to chime in if there is something else very specific that you'd like to see besides what I'm showing you. Something like an Eppendorf tube, if you want to label that. Obviously, Eppendorf tubes are very small. So in a procedure like this, it's quite small. So maybe all want to do is blow it up, add your text to it. This is very inaccurate, but I'm just doing it for demonstrative purposes. Or maybe you want to put it on the outside here and then Increase the font size. Yeah, by the way, BioRender has all of the usual text editing features you'd expect, like, changing the size, the font, line spacing, all that stuff. I also like to add in arrows. Oh, this is a little tip that I like to share. Try not to use arrows when you're labeling things. This actually is describing volume, 15 milliliters up to here. But if you're trying to label this line, sometimes it's better to actually use the arrow with the dot at the end. Where is it here? Where is the end? Here we go. The dot. And then I'm going to make this a little bit thicker. Width, I think, is what I want to change. Yes, width. That just really describes, that part of this object I'm labeling. Not sure if I answered the person's original question, but please let me know if I did. And then one little tip I'll leave you with, and I hope I'm not getting too technical here. Because Eppendorf tubes are pretty small, sometimes what I like to do is make a little circle crop to show that what I'm showing is actually not that size, but I'm zooming into it. What I mean by that is if my Eppendorf tube was really small on my diagram, I can actually use what should we call it? I think it's called a call out, maybe. Yes, here we go. Call outs, circles. This is very common. And a lot of science is describing things that are so small you can't see it, or so big it won't fit on your page. So sometimes you need these sort of tricks to be able to, highlight things that are not visible with the naked eye. And this I'm going to bring to the front because this is behind my circle here. Send to front. And then I'm going to circle crop this. Where is my crop? Oh, you know what? I might have to group this first. And then I'm going to crop it with a circle. And hopefully you can rewatch this recording if I'm going too fast, but I've heard feedback that this is a really neat feature that a lot of people like to use. So here we go, circle crop, enter. There we go. And I'll probably want to take this off and move it here. Okay. There we go. Anyway, just a small tip to show how you can show things that are really small, really big, without, keeping that Eppendorf tube like huge and in full visibility. You can actually crop in like a little window into like a microscope. Other questions? Microfluidic processes, yeah. And questions about ChemDraw for chemicals. Yeah, very good questions. We are thinking about an integration into a ChemDraw type of a library. So look out for that. We have some rudimentary chemistry structures. And some might be more like, individual elements like phosphate, for example, during a cellular process. Obviously, you need that. But we are going to be implementing more of like a simple, line diagram sort of structure, chem structure into BioRender. Great questions. I'm going to type in microfluidic into my search bar here, and you'll see that we've got a bunch of search results for that as well. Again, some are grouped icons. And going back to my search result, I think I have to like hit enter again. Yeah, there we go. Some are different, orientations. I usually like to look for ones that, again, are grouped icons so I can sort of manipulate those independently. This one, I can probably, change the position of these syringe tips. And this one, I can probably change, yeah, the neuron type or the color. Make it pop a little bit more. There we go. Oh, and this is neat. This icon looks like a layered icon. So I can change the color of the chip, the channels, and the wells. Let's try that out. Ooh, that's nice. Channel. Pretty cool. Yeah, so try that out. I just wanted to, again, really drive home the fact that some of our icons are really nicely editable like this. And hopefully, depending on your own field of research, we've got icons ready for you to use for these purposes. Awesome. Okay. Anything else? I wanted to leave you with a few last tips here because I think the power of BioRender is definitely in its illustration capabilities and fast figure making capabilities, but it's also more recently what I'm hearing is in its ability to collaborate with your teammates. So you can share the wealth and also share the burden a little bit. So if you want help in making your figure, or if you want feedback on, how your figure is looking, or, is this protocol ready to export? Can you come in here and add comments? Instead of your usual, printing it out or emailing, sending it to your PI or your student, having them print it out, mark it up with a marker, scan it, attach it, send it back and forth, back and forth. You can actually just share this as if it was a Google Doc. It is very secure, so you can only share it with people that you specify. So you can add your teammates. Maybe I'll add myself, but imagine that this is somebody else. I've sent the invite. So now there are 2 people on this document. And I close it up. And you can see here now, the other Shiz Aoki has access to this file. And what will happen is you can kind of come in here, change this to a purple fluid or something like that. And now I'm going to get a notification that there is a comment. If you're using slides, actually, it'll show the exact slide that the comment is on. So that's really neat as well. And eventually you'll have a diagram full of hopefully comments with really good feedback from your colleagues. And you can write done, post that, you can resolve the comment. But this really improves the efficiency of your workflow if you don't have to again, export, attach to an email, send it back and forth. If your person that you're tagging doesn't have a BioRender account yet, they can actually create one for free and start to contribute to your doc that way as well. So that's a really important feature that I think is definitely worth noting. It's a little bit unknown feature in BioRender that I think is quickly becoming a popular one. Another really helpful feature is this help button, and that is if you're in a jam and you can't find, for example, how do I change colors in BioRender, or maybe how to upgrade your subscription, how to request a custom icon, things like that. It's sort of your what used to be Microsoft Clippy. It's like your little go-to search tab. So if I type in changing colors, how do I use the color pick tool? Lots of different ways to get your answer. How do I change the canvas color? And what will pop up is sort of a little tutorial on how to change the canvas color, which is what I picked here. And a little GIF on how to do that. Okay. So that's there for your resource if you are stuck on how to use certain features in BioRender. Oops, I'm going to close this up. And sometimes if you really can't find what you're looking for, you can actually send us a message and start to chat live with our very knowledgeable customer experience team. Usually between the hours, we operate on Eastern time, so it's got to be around 9 to 6 Eastern, Monday to Friday, sometimes on weekends. But usually we'll have your answers sort of in a pre-written article. All right, we covered a lot, but still had so much to go through because there are a lot more use cases in BioRender. We hope that you have fun making your first figures, your first posters, your first slide decks within the app. We'd love to hear feedback if you have it, and hope that this tutorial was helpful. Thank you so much for joining. Happy to stay back for a few minutes to answer any more questions you might have. I'm going to wrap up here by showing a little bit of our sort of folder system and how to organize your files. once you have, hundreds of figures in your BioRender account, you might want a way to organize it better. And really easy to do that. Um, I can drag and drop files like this right into a folder to stay organized, like so. And if I click into that, obviously those files will be visible there. Gonna just navigate over to the chat to see if anybody has any last questions. Awesome. Yeah, a lot of questions about whether we'll expand the icon offerings to other science fields, and it's always on our roadmap or our wish list to do so. And our team is working vigorously to keep up with everyone's requests. So keep that coming. In fact, we do continually accept requests for templates or icons. In fact, here's our BioRender template gallery again, and this big button, I think it shows up intermittently, but if you have requests for a specific template type, we love fulfilling those if we have the bandwidth or if enough people request it. It does float to the top as priority. So every week, if you come back here, we've got a ton more templates added to this gallery. And again, that's the beauty of BioRender and for sticking around with us month after month is that it continually improves and you'll see changes and more features and more content every time you come back. Okay. Awesome. Okay, that was a lot of information. So maybe I'll pause there. But again, feel free to write into support at BioRender if you have any feedback or questions. We'll send a recording of this to the email that you signed up for the webinar with, so you can rewatch any part of this. And I encourage you to join maybe some future BioRender 101s, because depending on the person that runs it, you might hear a little bit different insights, different questions asked from the audience. And then we also run specific webinars, how to make a better grant figure, how to make a better presentation slide, all of those things. So if you sign up for our newsletter, you can stay in the know and join some of our more topical webinars that way. Awesome. Okay. I guess we'll sign off here, give everyone a few more minutes back in their day. Thank you so much for joining, and good luck with your figure making. We can't wait to see what you create.